put them back in there.
No, you can leave them out.
Okay.
Hi, Hannah. Hi. So I've been making fabric masks for the hospital in Peterborough because they put out a call for them. So I made some with ties. And then I made other ones that have elastic around the ears, but I ran out of elastic and I can't find any anywhere. So.
The ties are way better in my opinion.
You think so? Yeah.
Yeah, elastic hurts your ears.
I know. Well, the other thing is it has to kind of be fit to you, right? Because, you know, on one person, it's going to be too loose. So I don't know. I made both kinds. Like, I don't like. The other thing I did was I got some, I don't know if you've read about this, but some people are making masks out of vacuum bags that are HEPA grade. You know, they have HEPA filter in the bag. I may try my hand at those. We'll see. I just want to do something. I feel like it feels very odd not to be doing something to help. You know what I mean? So. All right, well, I imagine a couple people, I know a couple people are just finishing up the exam. So we'll let them kind of join in as they come. Remember, I'm gonna go through and grade all of the exams by hand, and I'll, you know, I'll look at the bonus question and all that too. So just stay tuned, and pretty soon you'll get to see your final score and what you might have missed, what you got correct and what you didn't get correct. So as soon as those are ready. What we're going to be doing today is our last metabolic exercise, which is respiration. And this exercise isn't too bad. It's not particularly long. It starts in the lab manual on page 139. Just like we did last time, we're going to talk a little bit about the tests that are introduced in this lab. And I'm going to tell you what questions that I want you to answer in your lab manual and which questions you can just cross out. The other thing I want to mention is I have posted a revised schedule for us up on the homepage on Canvas. I made an executive decision that we're going to do a modification of the unknown microbe exercise. So you are not going to be writing a lab report for me for the unknown microbe exercise. There is an exercise, a last exercise in your lab manual. If you turn to page 147, and it's called Unknown Identification and Berge's Manual. And in this, they talk about how we use what are called dichotomous keys and flow charts and things like that in the laboratory when we're trying to identify a microbe. How we do all of our testing, our gram staining, and all of our metabolic testing, and we look at colony morphology and things like that. And we use all of that data in those tables, in those keys, to help us identify the organism. So next time when we get together, we're going to talk about that exercise, what I'd like you to do for that exercise, but you won't be writing a lab report for me at the end of the semester, okay? And again, you can see the revised schedule up on Canvas with all of the revisions to when you're going to need to get your lab notebooks to me and so on, okay? But what I'd like to do now is talk a little bit about how we test for respiration in the laboratory. So if you remember, as you were just taking your test, you were tested on this. Remember that when we talk about cellular respiration in microbes, we're talking about this long series of oxidation reduction reactions, with oxidation being the loss of electrons and reduction being the gain of electrons. And we also talk quite a bit about how diverse microbes are in their metabolic abilities. You know, some of them do aerobic cellular respiration, and some of them do anaerobic cellular respiration. They make ATP through oxidative phosphorylation. They can also use fermentation and substrate level phosphorylation and all kinds of combinations of those. So they're very diverse. And so it's important for us to know, in terms of respiration, what their abilities are. So I want to talk for just a second about aerobic organisms that are doing cellular respiration. Remember, they're using oxygen as their final electron acceptor molecule. And that means that that oxygen is getting reduced. If you start at the beginning of respiration, glucose is getting oxidized. And then at the end of respiration, all the way at the end, oxygen is getting reduced. So that oxygen gas is accepting electrons from the electron transport chain, and it's actually becoming water. Now, something that we didn't talk about in terms of the electron transport chain is that there's a molecule in there called a cytochrome. And cytochromes are involved in passing the electrons down the electron transport chain. In bacteria, there are actually 4 classes of these molecules called cytochromes. And lucky for us, they're named A, B, C, and D. There's one kind of cytochrome called cytochrome C that's actually quite common in bacteria, and it's one of the things we test for metabolically. We look to see if they have cytochrome C, because that's one way for us to know that they're doing this aerobic respiration. So when we think about anaerobes, it's a little bit different. Remember, anaerobes aren't using oxygen at the end of their electron transport chain. They're using a different molecule. And we said some of the most common ones are nitrate and sulfate and carbonate. One of the things we didn't mention were names of other molecules that anaerobes use at the end of their chain, but there are a lot of them. I've got some of the organic molecules listed here as well, meaning carbon containing. A lot of them use the ones we learned, which is nitrate, sulfate, carbonate. I just want you to know that there are others as well. Now, when we talk about nitrate as the final electron acceptor, we're talking about one of the most common final acceptors that you see in bacteria. So what I have on the slide here is a description of what happens to the nitrate. Remember, we said for an aerobic organism, the oxygen at the end of the electron transport chain is going to get reduced and become water. For an anaerobic organism that uses nitrate, again, one of the most common final electron acceptors, that nitrate is going to get reduced. But nitrate, when it takes on electrons, it becomes nitrite. And that's what NO2 minus is, nitrite. But it doesn't stop there. Of course, microbes, remember, are incredibly diverse. So while some of them use their nitrate and it gets reduced to nitrite, some of them keep going with it. They then take the nitrite, the NO2, and it accepts more electrons. And it creates a molecule called nitrous oxide. There are some microbes that go even further. They'll take their nitrous oxide molecule and they'll feed it even more electrons. And that will create nitrogen gas. So when it comes to anaerobes who use nitrate as their final electron acceptor, they actually differ in what they do with the nitrate. Some of them turn nitrate to nitrite. Some of them turn nitrate to nitrite to nitrous oxide. And some of them turn nitrate to nitrite to nitrous oxide to nitrous gas. And the only difference between those pathways is how many electrons are being fed in. In other words, how extensive the reduction is of the nitrate. I also wanted to point out that these equations that you're looking at on this slide are not balanced. They're not balanced. So I just want you to know what's going in and what's coming out. Now, remember the other two very common final electron acceptors in anaerobes are sulfate and carbonate. So I've also included the equations for sulfate and for carbonate, and they're much simpler. The sulfate molecule, when it accepts electrons, when it gets reduced, becomes hydrogen sulfide. And we talked about how we can look for hydrogen sulfide in media, in agar and in broth. We can test for the presence of hydrogen sulfide. The other one, the carbonate, which is CO3 minus 2, when that one gets reduced, when it accepts electrons, it becomes methane. So that is one of the gases that we can test for, we can look for in, again, in broth or in other test media. So we're going to be looking at 3 different tests today that are used to assess the type of respiration that a microbe does. We're going to be doing the oxidase test, the Catalase test, and what's called the nitrate reduction test. Now, one of these you've already looked at. Remember, we practiced the Catalase test in lab. We said that the Catalase test is used to test for the presence of an enzyme called Catalase. And if you remember, Catalase is used by a microbe to break down one of the toxic forms of oxygen, which is hydrogen peroxide. So if a cell has Catalase, it can break down hydrogen peroxide into oxygen gas and water. And if you remember from lab, when you do this test, you take some hydrogen peroxide and you put it on top of the colony material and you look for bubbles. Because if there's catalase in those cells, they are breaking down the hydrogen peroxide and they're liberating oxygen gas. That's what the bubbles come from, the oxygen gas. So the catalase test can be used, metabolically speaking, to identify different types of organisms, because we would expect, if you recall, aerobic organisms, all of the different classes of aerobic organisms. Remember, some of those are obligate aerobes, some of them are micro aerophiles, some of them are aerotolerant, and so on. We would expect them to have catalase if they're getting exposed to oxygen, if they're using oxygen metabolically, in other words. We would expect catalase to be present. The only ones that we don't really expect to have catalase are the true anaerobes, because they're not using oxygen. Obligate anaerobes have no reason to have oxygen around. And so they are less likely to need catalase enzyme available because they're less likely to be exposed to hydrogen peroxide. Now, the second test on the list is this oxidase test. And what the oxidase test is testing for is the presence of that molecule called cytochrome C. Remember, this is a molecule that sits in the electron transport chain, but it's not part of the chain. The chain is actually proteins, big groups of proteins. And if you recall, the electron transport chain is going to pass electrons from place to place down the chain. So what cytochrome C does is help that happen. Cytochrome C basically accepts electrons from some of those proteins and then passes it along to other proteins in the electron transport chain. In the oxidase test, we work with this chemical called oxidase reagent. And what we do is we apply the reagent to some colony material. Now, this reagent, when it is in the presence of this cytochrome C, it creates a molecule called indophenol blue. And you guessed it, indophenol blue has a color. It's not blue though. It's actually a purple color, and it really is a purple. It's a true purple color. So a positive oxidase test produces a very visible blue color. And we'll take a look at that in just a minute. The last test that we're going to look at today is a nitrate reduction test. Now, this test is quite complex because it has several steps in it, and you have to be able to interpret the results of each step. But again, it's very useful to us when we're trying to identify an organism, a bacterium, because it gives us a real insight into how they metabolize and if they use nitrate in their cellular respiration process, if they use it as their final electron acceptor. The nitrate reduction tests tells us how far they're going to reduce the nitrate. Remember, some organisms reduce it a little, and some organisms reduce it a lot. Some organisms only take on a few electrons per nitrate and create nitrite, and other organisms take the nitrite and keep reducing it. They make nitrous oxide, and then from the nitrous oxide, they can even make nitrogen gas. So this nitrate reduction test is going to allow us to find out just how far they reduce their nitrate. Now, it uses a broth. It uses a very, just a simple nutrient broth, and it adds a source of nitrate into the broth. It's typically a potassium nitrate. You inoculate the broth tube, and then you put it in the incubator for a full 48 hours. Remember, when we do these kind of long incubations in the incubator, it's because it takes time for these metabolic reactions to occur. Now, again, this test involves sequential reagents. So the first thing we do when we take the tube out of the incubator is we add 2 different reagents. And I have the names on this slide here, but I don't want you to worry about them too much. The first one is called dimethyl alpha naphthylamine, and the second one is called sulfonylic acid. Now, when you buy this test commercially, the test refers to the dimethyl alpha naphthylamine as reagent A, and the sulfonylic acid is called reagent B. So again, I'm not going to make you learn the names of these 2 reagents. Just know that there are 2, and we put them both into the tube when we take it out of the incubator, and we look for a red color change. If the broth turns red, what that tells us is that the organism growing in that tube has taken the nitrate and used it at the end of its electron transport chain, and that nitrate has accepted electrons. It's been reduced, and it has become nitrite. So again, when a red color change is found after the first 2 reagents are applied, that's a positive test. That's a positive test. We can say that that organism reduces its nitrate. And we can also say that it reduces it to nitrite. Now, what do you do if you don't get a red color change? If you think about it, one of 2 things could be going on in that tube. The first thing that could be going on is that the bacteria doesn't use nitrate at the bottom of its electron transport chain, right? That's the first thing that could be going on. The second thing that could be going on is that it's reducing nitrate. It's using it, but it's reducing it beyond the nitrite stage. It may be going from nitrate to nitrite to nitrous oxide, and even all the way to nitrogen gas. So if you don't get a red color after you put the first 2 reagents in, you still don't really know the whole story. So what you do at that juncture is you sprinkle in another reagent, which is a powder made out of zinc. You just sprinkle in a little bit of this zinc powder. If there's any nitrate in that tube at that moment, the zinc will reduce it to nitrite. All right, let me say that again because that's really important. If there's any nitrate in that tube in that moment, and that would mean that the bacteria does not use nitrate at the end of its electron transport chain. If there's any nitrate in that tube, the zinc will reduce it, and it will produce nitrite, and the tube will turn red. So after you put the zinc in, if the tube turns red, you've got a negative reaction for the nitrate reduction test. It's not the bacteria that reduced the nitrate. It was the zinc that reduced the nitrate. Now, you still have one more step. Let's say you take the tube out of the incubator, you put in reagent A and reagent B, and you get no red color. So then you do your second step and you sprinkle in some zinc powder, and you still get no red color. Now what do you do? Well, after the zinc reagent is added, if there's still no red color, it means that there's no nitrate left in that tube. So it's a positive test for nitrate reduction. You just need to find out if the bacteria is reducing the nitrate to nitrous oxide or nitrogen gas. And the way you do that is by inserting one of those little upside down test tubes into the tube, that Durham tube that we saw when we did carbohydrate fermentation. If the bacterial cell is making nitrous oxide from the nitrate, there will be no gas in the Durham tube. If the bacterial cell is making nitrogen gas from the nitrate, you'll have gas in the Durham tube. So wow, what a complicated test, right? Now I want to take a minute and just look at these tests and what a positive result looks like. We'll start with that oxidase test. Remember, the oxidase test is testing for the presence of cytochrome C. So an organism, a bacterium that has cytochrome C has an electron transport chain. And if the cytochrome C variety is in the electron transport chain, you'll get a positive oxidase test. Now, there's a couple ways you can do this test. If you look at the slide that I'm going to show you right now, in the picture with the 2 cotton swabs, what you do is you take the cotton swab and you pick up a few colonies off an agar plate. And then you sprinkle onto the colonies a couple of drops of this oxidase reagent, as it's called. If that organism has cytochrome C in it, you'll get a purple color. And it really is just like this. It is a blatant positive result. You have to read this. You have to read this quickly, though. You have to read this test very quickly, just like the Catalase test. You have to read it immediately. Now, the other thing, the other problem with doing the oxidase test using a cotton swab is that the oxidase reagent is very labile. It means that once you open it up, it doesn't last very long. So, you know, when we're doing a lab with a bunch of students and we can go through the entire tube of oxidase in one lab period, it doesn't matter. And so we commonly do it in lab using cotton swabs. But if you're in a commercial lab and you have a whole container of oxidase reagent and you don't want to waste it on 1 or 2 tests, there are other ways to do the oxidase test. You can purchase these little strips that say oxidase on them. These strips have the oxidase reagent impregnated in them. So all you have to do is pick up some colony material and just smear it on the strip. If you get purple where you've smeared the colonies, that's a positive test for oxidase. And if you don't get purple, it's negative. These strips are really inexpensive and you don't waste oxidase reagent like you do when you use the swabs. The last thing you can do, although I've personally never done it this way, you can take your whole plate, you can take your whole agar plate and you can drip the oxidase reagent directly onto the colonies. It apparently works. You'll get a purple color, but it has the same drawback as the swabs in that you're going to need to open a bottle of oxidase reagent. And once you open it, it has a very limited shelf life. So I don't know anybody who does it this way, but it is possible to do it this way. The strips are really the most efficient way and the most cost-effective way to do the oxidase test. You've done a Catalase test. You did it this way on this slide, where you took some colony material and you put it on a glass slide, and then you put hydrogen peroxide on top of it, and you watched for bubbling. Remember, the bubbles are created because the catalase enzyme is breaking down the hydrogen peroxide, and it's making oxygen gas in the process. You have to read these quickly. And the other thing to remember, the critical thing to remember, is you can't put the colonies on the slide with a metal loop. I don't know if you remember that part, but you have to use a wooden cotton swab to transfer the colonies, because aluminum metal will give you a false positive catalase test. I also included this picture over on the right. It is possible to do a catalase test with a broth culture. All you have to do is take a little bit of the broth and just drip some hydrogen peroxide in there. You will still see bubbling. You'll get some bubbling in the tube when you have a positive catalase result. Now let's look at the nitrate reduction test. And remember, this one has steps in it. You have to add reagents in sequence. This is the color change over on the right-hand side of this slide. This is the color change that you're looking for at each step. On the left here, you can see a test tube with broth in it. So we inoculated this tube with cells and we put it in the incubator. 48 hours later, we took it out. We're looking for a color change, and it's a blatant color change. It's red, okay? So if you see red, you know you have nitrite in that tube. So when you take the tube out, it's going to look like this one over here on the left. It's going to be a clear color, or the color of the broth. And remember, the very first thing you do is you put in 2 reagents. You put in reagent A and reagent B, and you look for the color change. Now, if you go to the flow chart over here on the left-hand side of the slide, you can see one of 2 things is going to happen. You're either going to get a red color or you're not. If you get a red color, you can say that that is a positive nitrate reduction test, because the organism has turned nitrate into nitrite. And it's the nitrite that's giving you that red color. If you get no color change, you got to keep going, because you're not quite sure yet what's happening in the tube. If you get a colorless reaction, you add the zinc in. And one of 2 things is going to happen. Either it will turn red or it won't. If it turns red, remember, that means that there's nitrite in there, but the bacteria didn't make that nitrite. The zinc did. So if you get a red color after you put the zinc in, that's a negative nitrate reduction test. The bacterial cell didn't reduce the nitrate. It can't. It doesn't use it. The zinc did it. The zinc turned the nitrate to nitrite, and you got the red color. If you don't get a red color change after you put the zinc in, now you have to look in the Durham tube. If there's no gas in the Durham tube, that means you got a positive nitrate reduction test, and the organism reduced nitrate to nitrous oxide. Let me say that again, because that's important. If there's no gas in the Durham tube, you have a positive nitrate reduction test, and your organism reduced nitrate to nitrous oxide. If you do have gas in the Durham tube, you have a positive nitrate reduction test, and the organism reduced nitrate all the way to nitrogen gas. So looking at the flow chart, this is a positive test. This is a negative test. And this is a positive test. It's just that we went beyond nitrite all the way to either nitrous oxide or nitrogen gas. So yeah, the nitrate reduction test, you got to put your thinking cap on when you look at your results. Take a look at this, this next slide, and you'll see some results from a nitrate reduction test. And they've got the tubes labeled as to which organism is inside. You've got a strep pneumonia over here on the left. A staph epidermidis is next. Then E. coli. And then on the right is Pseudomonas. And they've got the tubes labeled as to what sequence of events has happened to them. So over here on the left-hand side in the Streptococcus pneumonia tube, it says it turned red after the zinc was added. Now, using the flow chart on the last page, what do you think that result is? Take a look at the flow chart. Sorry, let me show it to you again. For that strep pneumonia bug, they got a red result after the zinc was added. Now, is that a positive nitrate reduction test, or is that a negative nitrate reduction test? This is after the zinc was added. I had a couple votes for positive. Anybody else want to join in? Excuse me. Okay. After the zinc is added, it's a negative test. I know, it's confusing. Let's go through it again. When the tube comes out of the incubator, the first thing we do is we put in reagent A and reagent B. If the tube turns red, that's a positive test. That means that the bacteria reduced nitrate to nitrite. If we don't get a red color after we put in reagent A and reagent B, we sprinkle in a little bit of zinc. If the tube turns red then, after the zinc goes in, it means that the bacteria didn't use the nitrate. The bacteria did not reduce nitrate to nitrite, but the zinc did. The zinc took the nitrate that was still sitting in that tube, because the bacteria didn't use it, and it reduced it to nitrite, and it turned the tube red. So when the zinc goes in, if you get a red color, you've got a negative nitrate reduction test. The bacteria did not reduce the nitrate. The zinc did, though. Now, let's take a look at those results again. Look at the two tubes in the middle, Staphylococcus epidermidis and E. coli. And I realize that these have more of an orangey color to them, but we're going to interpret that as red. And you can see the label underneath says turned red after reagent 1 and reagent 2 were added. So these two tubes came out of the incubator. We put in reagent 1 and reagent 2, and they turned red. Is that a positive test or a negative test? What do you think? Positive. Positive. Yeah, you're right. If you put the reagents in, reagent A and reagent B, or reagent 1 and reagent 2, when the tube first comes out of the incubator, and you get a red color, it means that the bacterium took the nitrate and reduced it to nitrite. That's what you're seeing. You're seeing nitrite in that tube. Now let's look at the tube that's over on the right-hand side, the Pseudomonas tube. This one stayed colorless after the zinc was added. It stayed colorless. So first of all, is that a positive nitrate reduction test or a negative nitrate reduction test? So this Pseudomonas tube came out of the incubator. We put in reagent 1 and reagent 2, and it didn't turn red. So then we sprinkled in some zinc dust, and it didn't turn red. Positive or negative for nitrate reduction? Positive. Good. Good. Yeah, this bacteria, this Pseudomonas, absolutely reduced the nitrate. It's just that it didn't stop at nitrite. It kept reducing. It took the nitrite and it reduced it even further. Now, how do I know how far it took it? What do I have to check in that Pseudomonas tube? What do I have to look at now? In this Pseudomonas tube, what do I do? What do you think? What do I have to look at in this tube? It's not red. What else should I look at? That's right, Hannah. We're going to look in the Durham tube. We're going to look for gas in the Durham tube. This is a positive result for nitrate reduction.